Welcome to Section 2 of Microbiology Fundamentals. In this section, we'll be discussing virulence. Let's get started. Virulence describes the bacteria's ability to do three things: invade the host, evade the immune defenses, or inflict disease. And virulence is achieved through virulence factors. And this table lists the virulence factors that you need to know, as well as the action of the virulence factor, this middle column. And lastly, how this virulence factor plays a role in virulence. Now, this table does not provide the details you need to know about each virulence factor. I simply show this to you here so you can briefly see the roles various factors have on virulence. When you look at these top three, you can see that they increase virulence by helping the bacterium invade the host. The next three increase the virulence by allowing the organism to evade immune defenses. And the bottom three increase virulence by allowing the organism to inflict disease. Many of these items have been discussed in detail in the previous video on bacterial structures, for example, pili. In terms of virulence, these pili allow the organism to bind to host surfaces and invade the host. Flagella allow the organism to move around and ultimately allow it to invade the host. And the capsule prevents phagocytosis, which allows the organism to evade immune defenses. And as I mentioned, these three items were discussed in great detail, the detail that you need to know for step 1 in the previous video. The remainder of the lecture will be focused on the details of these other items that were not previously covered. For example, IgA protease, M protein, protein A, the type 3 secretion system, endotoxins, and finally exotoxins. So let's dive into IgA protease. IgA protease cleaves IgA antibodies. IgA antibodies normally defend the mucosal surfaces, so bacteria that can cleave these antibodies make it easier for the bacteria to adhere to mucosal surfaces. And IgA protease is secreted by multiple bacteria, for example, Strep pneumoniae, H. flu type B, Neisseria meningitidis, and Neisseria gonorrhea. So again, IgA protease allows it to adhere to the host, which means it's allowed to invade the host, which is why it's virulent. Now let's discuss M protein. M protein is considered a virulence factor because it prevents opsonization and phagocytosis. In other words, the M protein is considered a virulence factor because it allows the organism, the bacteria, to avoid immune defenses. Now, an interesting thing to know about the M protein is that it's similar to the antigens that you have in the heart. So, when antibodies are formed against the M protein, those antibodies can then attack the heart as well. And this is what causes rheumatic fever. So in other words, M protein causes the body to attack itself and create this disease. And the organism that uses this is Strep pyogenes, or group A strep. Next, let's discuss protein A. Protein A is something that binds to the Fc portion of IgG antibodies. And this means that only the FAB portion of the IgG antibody is exposed. And this prevents opsonization and phagocytosis of the bacterium. And protein A is on the surface of Staph aureus. Now this idea can be a little bit confusing, so we've got a diagram to help you understand it. The top image depicts the normal process. Here is an invading bacteria, and antibodies, which you can see right here, recognize an antigen on the bacteria. And the portion that recognizes the antigen is the FAB portion of the antibody. This leaves the Fc portion exposed, and it's this Fc fragment that receives the macrophage. The macrophage comes and it binds to this Fc portion, thus leading to phagocytosis and death of the pathogen. If you look on the lower part of the image, we can see what happens when protein A is involved. And this is used by Staph aureus. Staph aureus will come in, and it has protein A on its surface. It will bind to the Fc portion of the antibody, and therefore the Fc portion isn't exposed and available for the macrophage. And this means that the macrophage cannot bind to Staph aureus. Thus, protein A allows Staph aureus to evade the immune system. Now let's do a question to review what you've learned so far. A bacterial organism produces a virulence factor that interacts with host antibodies, allowing it to adhere to host surfaces. Which of the following statements is consistent with this virulence factor? A. It is produced by Staph aureus. B. It is secreted from the pathogen. C. It can lead to rheumatic fever. Or D. It is part of the cell surface. Hopefully from the question stem you notice two important points. It interacts with host antibodies, and somehow that interaction allows it to adhere to host surfaces. This describes IgA protease. With that in mind, the correct answer is B. It is secreted from the pathogen. Recall from our discussion that IgA protease is secreted by multiple bacteria, and it cleaves the IgA antibodies. In other words, it interacts with those antibodies, and this allows the bacteria to adhere to mucosal surfaces, as the question suggests. Now choice A is wrong because Staph aureus does produce a virulence factor that interacts with host antibodies. This is called protein A. However, protein A isn't responsible for allowing the pathogen to adhere to host surfaces. Its role is to help the pathogen evade phagocytosis, And choice C is wrong because this describes the M protein. And the M protein is not responsible for allowing the bacteria to adhere to host surfaces, as the question stem suggests. And lastly, choice D is wrong because protein A is not part of the cell surface. It is secreted. Next, let's talk about the type 3 secretion system. The type 3 secretion system can also be called an injectosome, and the reason is because it's a protein structure that can insert into host cells. And when it does this, it can inject exotoxins directly into the host cell, hence that term injectosome. And this is used by several gram-negative organisms. For example, Salmonella species, Shigella species, E. coli species, and Pseudomonas aeruginosa. Here are two images depicting a type 3 secretion system. The left is a diagram showing its structure embedded in the envelope of gram-negative bacteria. And we know this is gram-negative because here's the outer membrane, something that's not present in gram-positive bacteria, which we discussed in the previous lecture. Up above, we can see the host cell membrane, and you can see the needle of the injectosome with that tip ready to inject or penetrate that host cell membrane, allowing it to directly inject its exotoxins. On the right, we see an image from an electron microscope, just showing these injectosomes to help illustrate how cool researchers are since they can somehow isolate these tiny structures to show us. Now, as we mentioned before, they're utilized by several gram-negative organisms, like these ones listed right here. Don't try memorizing how these organisms have a type 3 secretion system right now, because when you learn the specific pathogens in our bacteria mnemonics, we'll help you memorize that these bacteria use this type of secretion system. In other words, don't worry about this now. For now, just know that the type 3 secretion system is used in gram-negative organisms. Now that's all you need to know about the type 3 secretion system. We mentioned that it injects exotoxins. We will dive into the details of exotoxins during the last segment of this lecture. And don't worry, we're going to cover the details of exotoxins at the very end of this lecture. Before we get to that, let's discuss endotoxins. Endotoxins are lipopolysaccharides, or LPS, and these are on the surface of gram-negative bacteria, and lipid A is the toxic component of the lipopolysaccharide. This diagram was discussed in section 1 when teaching about the gram-negative bacterial structures. I show this to you again to emphasize that endotoxin is embedded in the bacteria, which you can see right here. It's not secreted. And this is a lipopolysaccharide, which is the endotoxin. And the toxic component of the endotoxin or lipopolysaccharide is lipid A. And when the cell dies, this lipid A gets released into the bloodstream. So again, when the bacterial cell dies, it will release lipid A, and this causes the host immune system to release lots of cytokines. For example, TNF-alpha, IL-1, and IL-6. This is bad because it happens all over the body, causing sepsis, or even septic shock. Now, sepsis and septic shock are important terms to be familiar with, so we're going to dive into those really quick. In the past, sepsis has had several terms surrounding it. For example, severe sepsis was a term. It's actually been phased out. In fact, in 2016, this special task force updated the terminology to basically two things that you need to know. There's sepsis and there's septic shock. It's that simple. Sepsis is defined as life-threatening organ dysfunction caused by a dysregulated host response to the infection. And septic shock just means that it's sepsis that requires a vasopressor to maintain adequate blood pressure. This slide demonstrates the basic mechanism of sepsis. In popular Step 1 review books, you may see complicated diagrams showing how gram-negative bacteria can lead to overactivation of the immune response. And in those diagrams, they detail every cytokine and how it contributes to sepsis. That kind of information is totally unnecessary in this context. I will show you how to diagnose sepsis and dominate your exams. Plus, diagrams like the one I described are not even unique to gram-negative bacteria, because sepsis can occur from several organisms. So instead of all that complicated material, here's what you need to know. A virulence factor triggers the immune response. This leads to organ dysfunction, and you can suspect that organs are going to be dysfunctional when you see that the patient has hypotension. Lastly, gram-negative bacteria, gram-positive bacteria, and fungi can all cause sepsis. But an important point to remember is that when dealing with sepsis in patients with gram-negative bacteria, your mind should go specifically to lipid A. In other words, a patient has bacteremia with a gram-negative organism, and then they have sepsis, like hypotension, you should think, oh, this is caused by lipid A. When dealing with sepsis in patients with gram-positive infections, or even fungal infections, your mind shouldn't be focused on any one virulence factor. After all, these organisms have many virulence factors, many of which can trigger a host immune response. I've included a reference to an article right here that can be super helpful on this topic if you want to go deeper. But what I've covered in this slide is sufficient for step 1. Now let's come back to finish our discussion of endotoxins. We just mentioned that they can cause sepsis and septic shock, but it's important to know that the source of these endotoxin genes are from the bacterial chromosome, which is fairly intuitive. It's where you would expect all bacterial genes to come from. However, It's important to know that endotoxin genes come from the bacterial chromosome when you start thinking about exotoxin genes, because exotoxin genes can and often do come from places other than the chromosome. But we'll get to that later. Now let's do another quick question to tie some of these items together. An 18-year-old female who has been feeling sick is brought to the emergency department by her mother. Upon arrival, her temperature is 39.4 degrees Celsius or 103.0 Fahrenheit, and her blood pressure is 78/45 millimeters of mercury. Blood cultures confirm bacteremia with strep pyogenes. Which statement is true regarding how the pathogen caused her septic shock? Hopefully from the question stem, you notice that the girl has a fever, and she has extremely low blood pressure. This indicates shock, and she has bacteremia. So before we answer the question, what's the mechanism for septic shock? It's dysregulated immune response to an infection, not a direct result of the pathogen. With this in mind, the correct answer is choice C. A virulence factor triggered the release of TNF-alpha. This is true. TNF-alpha is one of several inflammatory cytokines released by our immune cells, and the term virulence factor is broad enough to encompass virtually any aspect of the pathogen that triggers this dramatic host response, leading to septic shock. Now, choice A is incorrect because this implies that strep pyogenes release the cytokines, which is not true. We know that the immune system releases those. And choice B is wrong because lipid A is only on gram-negative bacteria, and strep pyogenes is a gram-positive organism. And choice D is incorrect because strep pyogenes does produce an exotoxin that can cause cellular damage, but this activity does not directly cause septic shock. Because remember, it's the immune system that causes the septic shock by releasing all those cytokines. So D is wrong. And the correct answer is choice C. As a side note, we mentioned that this patient has low blood pressure, hypotension, which indicates that she has sepsis. And the new terminology presented by that 2016 task force states that septic shock is sepsis that requires a vasopressor to maintain adequate blood pressure. So just seeing that she has low blood pressure doesn't necessarily mean that we're going to call it septic shock. But our question at the end states that this is septic shock. So we are to assume that this patient requires a vasopressor to bring her blood pressure up. Now let's jump into exotoxins, our final topic. These are secreted from bacteria, and their activities vary, and their role in virulence is to inflict disease. So exotoxins are just secreted proteins that inflict disease, and they're utilized by most gram-positive organisms and some gram-negative organisms, which is interesting because endotoxins were used only by gram-negative organisms. Anyways, the source of exotoxin genes are the bacterial chromosome, plasmids, and lysogenic bacteriophages. In the next lecture on bacterial genetics, we discuss plasmids and lysogenic bacteriophages in detail, so we won't dive into the details here. For now, just appreciate the fact that exotoxin genes can come from more than simply the bacterial chromosome. Now, understanding the mechanism of exotoxins is important and frequently tested on, and this table lists the exotoxin mechanisms you need to know. Don't freak out and try to memorize all these right now. I just want you to have a place where all the important details are consolidated. We will actually go through each mechanism visually, and all these details won't be overwhelming for you anymore. Now, there are exotoxins that are not listed on this table, but they are not listed on this table because the mechanism is not important for you to know, and you will learn what you need to know about those exotoxins as you move through these specific bacterial mnemonic videos. With that in mind, let's dive into the important exotoxin mechanisms, starting with the top 4. All 4 of these will kill cells by blocking protein synthesis. Explaining these ideas with a diagram is more sensible than reading these details to you, but these notes are here for your review. So let's show that diagram now. This diagram demonstrates protein synthesis in human cells. The process of protein synthesis or translation is described in great detail within the biochemistry chapter. As a brief review, when translating mRNA, elongation factors transfer amino acids from the P site to the A site, causing the peptide to lengthen. You can see that elongation takes place here. One of the elongation factors is elongation factor 2. With relation to elongation, there are 2 toxins to know about, diphtheria toxin and exotoxin A. These will block elongation factor 2, or EF2. This will cause chain termination. In other words, the peptide will stop growing. That means there will be lack of protein products in the cell, and the cell will die. Up here, we can see we have the ribosome. And the 2 toxins to know about with relation to the ribosome are the Shiga toxin and the Shiga-like toxin. The Shiga toxin coming from Shigella, and the Shiga-like toxin coming from enterohemorrhagic E. coli, or EHEC. These work by removing an adenine from the rRNA on the 60S ribosome, this large subunit right here. Attacking this prevents the formation of that ribosomal complex. This means that translation cannot occur. So, by blocking the 60S ribosome, there's a lack of protein products, and the cell eventually dies. An important distinction between these 2 exotoxins is that Shigella's Shiga toxin actually invades the GI cells, but the Shiga-like toxin does not invade those cells. It's just an important fact to remember. Regardless, both toxins block protein synthesis and kill the cell. Now let's talk about the next 4 exotoxins: the heat labile toxin, heat stable toxin, cholera toxin, and edema factor. All 4 of these will increase fluid secretion. The top 3 resulting in watery diarrhea, whereas the edema factor in anthrax just increases fluid or edema surrounding the black eschar in cutaneous anthrax. In general principles of physiology, G-coupled receptors were described using a diagram similar to this one, but this specific diagram demonstrates the mechanism of the heat labile toxin of ETEC, or enterotoxigenic E. coli, as well as the cholera toxin of cholera, as well as the edema factor of Bacillus anthracis. Here is the extracellular area, and here is the intracellular area. And the G protein coupled receptor is here. Well, the heat labile toxin and the cholera toxin will stimulate this G-alpha S subunit of the G protein complex. Once activated, this G-alpha S subunit will go and activate adenylate cyclase. Adenylate cyclase will convert ATP to cyclic AMP. The net effect of high cyclic AMP is to increase chloride in water in the lumen of the GI tract, as we've written up here. This ultimately results in watery diarrhea. Now let's move on to the edema factor again. In cutaneous anthrax, edema factor will actually behave just like adenylate cyclase and itself convert ATP to cyclic AMP. Instead of causing watery diarrhea, the cells in the skin just release water in the area around the black eschar in cutaneous anthrax. So up here we've shown how edema factor just causes edema around the black eschar. This diagram describes the heat stable toxin of enterotoxigenic E. coli, which you can see labeled over here. What happens is the heat stable toxin will bind to this receptor on the cell membrane, acting like a ligand. So it'll bind to this receptor and trigger intrinsic guanylate cyclase, which will cause GTP to be converted to cyclic GMP. And the net effect of all this increased cyclic GMP is decreased reabsorption of chloride and water in the gut, leading to watery diarrhea. So we just covered these 4 toxins. Now let's discuss pertussis toxin. This diagram describes the mechanism of the pertussis toxin, which you can see labeled right here. It acts by inhibiting the G-alpha I subunit. As a review, this G-alpha subunit can be an I or an S subunit, which we discussed before. Well, the I subunit acts to inhibit adenylate cyclase. You can see right here that inhibiting symbol. So you can think of pertussis toxin as inhibiting the inhibitor. So ultimately, pertussis toxin increases the activity of adenylate cyclase, which increases ATP conversion to cyclic AMP. The net effect of all this cyclic AMP is that neutrophils will be unable to be recruited to phagocytose the pertussis pathogen, so decreased phagocytosis. The exact mechanism for why increased cyclic AMP will lead to decreased neutrophil recruitment is well beyond the scope of board exams. Just make sure you know that pertussis toxin inhibits the GI subunit, which leads to decreased neutrophil recruitment. Now let's do a review question. A 30-year-old man with bloody diarrhea is diagnosed with a Shigella infection. Which statement describes the mechanism through which Shiga toxin alters host cell activities? A. Inhibition of an elongation factor. B. G-alpha S subunit stimulation. C. Ribonucleotide removal on rRNA. D. Increased levels of cyclic AMP. Now, Shiga toxin acts by disrupting protein synthesis. And recall there are 4 exotoxins with a similar mechanism. Shiga toxin, Shiga-like toxin, those attack the ribosomal subunit by removing rRNA on the 60S ribosome. And the other 2 toxins which inhibit protein synthesis are the diphtheria toxin and exotoxin A of Pseudomonas. In any case, we know the Shiga toxin mechanism, which makes choice C the correct answer, ribonucleotide removal on rRNA. Choice A is incorrect because this describes the mechanism of the diphtheria toxin and exotoxin A of Pseudomonas, which inhibits an elongation factor. Choice B is incorrect because this describes the mechanism of the cholera toxin and the heat labile toxin of enterotoxigenic E. coli, both of which stimulate the G-alpha S subunit, which then leads to adenylate cyclase overactivation, which then leads to overproduction of cyclic AMP and watery diarrhea. And choice D is also incorrect, because increased levels of cyclic AMP is part of that mechanism for causing watery diarrhea. Now let's discuss the exotoxins that mess with neurotransmission, tetanospasmin and botulinum toxin. This image depicts tetanospasmin in Clostridium tetani. When a foreign object punctures the skin, and you can see the skin here, and a foreign object puncturing it, then the organism vegetates at the wound site and produces tetanus toxin, which you can see here. The toxin then travels through the motor neuron from the periphery to the anterior horn of the spinal cord, which we've shown right here. This type of movement going from the periphery to the spinal cord is known as retrograde movement, which hopefully makes logical sense. If the movement were going from the spinal cord to the periphery, this would be known as anterograde movement. From the image, notice that a cross-section of the spinal cord is shown, and right at the anterior horn you can see the motor neuron and the interneuron, labeled here. The interneuron is also known as a Renshaw cell. Once the toxin reaches the anterior horn, it travels from the motor neuron to the Renshaw cell, and we can see that occurring right here. The toxin leaving the motor neuron and entering the Renshaw cell. Notice that within the Renshaw cell are vesicles containing the neurotransmitters GABA and glycine, which you can see right here. These vesicles normally interact with snare proteins, which you can see right here. The snare proteins are responsible for releasing GABA and glycine into the synapse, where the neurotransmitters can then inhibit the motor neuron. However, when tetanospasmin enters the Renshaw cell, it cleaves the snare proteins. You can see this activity right here. This prevents the release of GABA and glycine. So by decreasing GABA and glycine, the muscle becomes disinhibited, which is why the muscles are overstimulated and have increased muscle tone. This image is very similar to the last one, but it's altered to represent the botulinum toxin. We have the botulinum toxin entering the Renshaw cell, and notice we have the snare proteins here as well, and the botulinum toxin inhibits these snare proteins from releasing acetylcholine. Whereas before, when we were talking about tetanospasmin, the neurotransmitters were GABA and glycine. So here we're talking about acetylcholine. And when botulinum toxin inhibits the snare proteins from releasing acetylcholine, the motor neuron will not receive that stimulus from acetylcholine, which leads to flaccid paralysis. Now let's discuss the exotoxins that lyse cell membranes, alpha toxin and streptolysin O. This image shows the cell membrane of our cells. Here's the outside of the cell, and here's the inside. And it's separated by this cytoplasmic membrane, which you can see here. Clostridium perfringens uses this alpha toxin. This is simply a phospholipase which degrades the cell membrane, and it hydrolyzes lecithin. So you could also think of this as a lecithinase. On the right is the toxin produced by strep pyogenes, which is called streptolysin O. This also lyses cell membranes. One of the most important facts for you to know is that our body will form antibodies against streptolysin O, and we can actually detect that with serology. These are called antibodies against streptolysin O, or ASO antibodies. So when you're trying to diagnose rheumatic heart disease or rheumatic fever, or post-streptococcal glomerulonephritis, or even later acute pharyngitis, you can do a serological test to find these ASO antibodies. However, even though these ASO antibodies can be detected in rheumatic fever and post-streptococcal glomerulonephritis, it's super important to recognize that streptolysin O is not the cause of these diseases. For example, we know that rheumatic fever is caused by the M protein, which we discussed earlier. Now let's discuss these superantigens. This diagram demonstrates the activity of superantigens. Normally, a dendritic cell, which you can see here, will take in an antigen, right here, and then present it to a CD4 positive T cell, which is a Th1 cell. This Th1 cell will then become fully active and release cytokines, which you can see listed here. These include IL-1, IL-2, TNF-alpha, and interferon gamma. This is a normal response. However, in the case of superantigens, which we've shown right here, this antigen will basically lock the MHC class II and the T cell receptor, or TCR. The point is that this whole complex is basically locked, and there's a huge release of cytokines. This overabundance of cytokines causes a drastic immune response, leading to toxic shock. This occurs with two toxins, the toxic shock toxin, also known as TSST1, causing toxic shock syndrome, or the erythrogenic toxin A, which causes a toxic shock-like syndrome. So again, the top one is Staph aureus, and the bottom one is strep pyogenes. With strep pyogenes, also keep in mind that this toxic shock-like syndrome will also cause scarlet fever. So in addition to fever, rash, and shock, they'll have scarlet fever. Another interesting point is that patients with Staph aureus who are experiencing toxic shock are also likely to experience the effects of other staph exotoxins at the same time. For example, the effects of the exfoliative toxin. So a patient could have toxic shock from this exotoxin, and they can have a blistering desquamating rash from the exfoliative toxin. I also want to point out that in the absence of things like the exfoliative toxin, toxic shock syndrome also causes a rash, as I've listed here. However, it's not a blistering desquamating rash like that would be caused by exfoliative toxin. Now that we've covered the topic of virulence, let's do a review question to apply what you've learned. A 15-year-old male is infected with a bacterial organism that releases an exotoxin. The role of this exotoxin is to prevent the release of glycine in the synaptic cleft of neurons. This describes which exotoxin? A, alpha toxin, B, tetanospasmin, C, botulinum toxin, D, streptolysin O, or E, erythrogenic exotoxin A. Now, glycine is a neurotransmitter. Specifically, an inhibitory neurotransmitter. Therefore, preventing the activity of glycine would lead to overstimulation of motor neurons, or tetany. And the organism that produces this is Clostridium tetani. So the exotoxin we're dealing with is choice B, tetanospasmin. Recall that tetanospasmin prevents the snare proteins from releasing GABA and glycine. This leads to disinhibition and increased muscular tone. Choice A is wrong because alpha toxin is produced by Clostridium perfringens, and this lyses cell membranes. And this causes things like necrotizing fasciitis or myonecrosis, which is listed in your table. And this mechanism is similar to streptolysin O, choice D. And choice C is incorrect because botulinum toxin prevents the release of acetylcholine from the synaptic cleft. And without this acetylcholine stimulation, the patients will experience paralysis, not tetany. And lastly, choice E is incorrect because urethrogenetic exotoxin A is a superantigen that leads to toxic shock-like syndrome and scarlet fever.